get these whiteboard. So anyway, today we're going to talk a little bit more about taxonomy. We kind of left off talking about taxonomy and how we classify organisms. We discussed the branch of science that deals with naming and classifying organisms as taxonomy. And we said that scientists basically look at similarities and differences in order to assign organisms to various classifications. We said that the system that we use now for naming an organism is referred to as this binomial system of nomenclature. All right. This binomial system of nomenclature is a system in which each organism is assigned a two-part name. And we designated some rules for this name, right? So we have the genus and the species. So we went down that hierarchy of classification from domain, kingdom, phylum, class, order, family, genus, and species. When we get down to the genus and species, now we're looking at organisms that are pretty much the same, right? And so that first part of the name, the genus, there is a rule that has to always be capitalized, right? So like think about when you write like your first and last name, we capitalize them, right? It's a little bit different convention for organisms. We capitalize the first name, the first part of the name, the genus, but we do not capitalize the species. That is absolutely important that you write that down and make a note of that, because for the rest of your life, for the rest of the continuation through this program, all of the higher biology level courses that you take, your professors are going to expect that you understand rules for nomenclature, right? The genus is capitalized, the species is not. Both parts of the name are in italics when you're typing on your computer, when you're using a word processor. This name is always italicized. The name of an organism is always italicized, okay? If you're writing manuscript with your pencil, you underline the name, right? So these rules are going to be very important, and we'll definitely have exam questions to make sure that you can kind of identify which names are written correctly and which ones are not. So we capitalize the genus, the species is lowercase, the whole entire name is in italics, okay? We define the species as a group of organisms with very similar structure, function, and behavior, all right? So again, as we move down that hierarchy of classification, when we get to the species, these are organisms that are pretty much going to be identical, all right? And so there's one inclusion criteria for identifying a species, members of the same species, is that they consist of one or more populations whose members are capable of breeding and producing fertile offsprings, right? So if you think about the human species, right, we only breed and produce fertile offspring with other humans, okay? There are various populations of humans, but we're all humans, all right? And so that definition should sort of ring a little intuitive, right? Like humans, we are, for the most part, similar in structure, function, and behavior, all right? We did pause and acknowledge that there are anomalies, right? There are individuals who are born with various defects, if you will, but as a species, we have the same structure in terms of limbs, eyes, nose, so forth and so on, function, behavior. We pretty much behave the same as a species, right? I think this illustration of butterflies does a great job of highlighting that for the sake of defining a species, these are organisms with very similar structure, function, and behavior, but obviously there can be variations, as you see here with the wing color, wing span, wing length, that type of thing, but for the most part, the structure is the same. We can look at this picture and all acknowledge that these are all butterflies, right? And so we see that in all organisms that are assigned to the same species, all right? The inclusion criteria here is that organisms of the same species are going to be populations whose members are capable of breeding together and producing fertile offspring. So that is important. That should be in your notes already. I think this is kind of where we left off, where I was kind of rushing right at the end, was the 8 major categories of classification, with the species being the most specific. So I said that scientists are assigning organisms to these classification categories based on similarities in structure and things that we can see. And we also can use genetic analysis for other phylogenetic relationships that we want to identify. But structurally and so forth, we're looking for similarities, right? And as we're identifying, okay, is this a mammal? Is it, you know, warm-blooded, cold-blooded? Does it have, you know, 4 legs or 2? You know, there's different inclusion criteria that we use for each of these classification categories. And so as we move down this hierarchy, right, from domain, kingdom, phylum, class, order, family, genus, and species, the species is going to be the most specific, right? They're going to be very specific inclusion criteria as you move down this classification scheme, okay? And so as you move down this classification scheme, we're saying that organisms are going to increase in similarity, but decrease in number. So make sure you understand that. We're going to increase in similarity as we move down, but we're going to decrease in number. So we're saying that when we get down to the level of the genus and species, there are going to be fewer and fewer organisms included at this level of classification. So the species level is the most specific. At the domain level, this is the most broad level of classification. We've got domain, prokarya, eukarya, and archaea. Every single organism that has been identified can be categorized into one of these classifications, okay? Prokarya, eukarya, or archaea. So because of that, because there's 3 domains and there are millions of organisms, you know that this is going to be a very broad level of classification, okay? Our inclusion criteria here is the presence or absence of that nucleus, right? If it has a nucleus, what do we call those organisms? At the domain level. If it contains a nucleus, what are those called?
Prokaryote.
If it contains a nucleus.
Eukaryotes.
Eukaryotes contain a nucleus. Prokaryotes do not contain a nucleus. And then we highlighted that archaea are also a type of prokaryote, but they have some distinct differences that require them to have their own domain. All right. If you missed this conversation on Friday, quickly put in your notes that archaea are a type of bacteria. They're very similar to bacteria in terms of structure and so forth, but they share some very distinct physiological features that distinguishes them from being categorized as bacteria, right? They are able to survive in some very hostile environments that don't support normal bacteria life, right? So archaea are in their own domain, but they are prokaryotes. And so from domain, we went down to the kingdom and we highlighted our 5 kingdom system of classification. Can someone remind us of our 5 kingdoms really quickly? What are the 5 kingdoms for classification? Somebody.
Fungi, protists, plants, dang, I don't know how to say the word.
Fungi.
We got fungi, we got protista, we got plants. We're missing 2.
Animals.
Animals and
Bacteria.
bacteria.
Plants.
Bacteria, and it's M. You see the Monera. Monera is for bacteria. All right. So we got Plantae, Animaliae, Fungi, Protista, and Monera. Those are our 5 kingdoms, guys. You should certainly learn those. All right. And so then I mentioned that after that kingdom system, there isn't consistency after that in terms of name and number, right? The number of phyla, class, order, family, genus, and species will differ depending on the actual organism. All right. So I showed you this schematic already, the 3 domain systems, we have our 5 kingdom system, and then if we continue to move backwards, we've already sort of mentioned this theory of evolution that we've all derived from a common ancestor, right? So we do study phylogenetics and look at evolution over time, and we can trace back these common ancestor origins, and look at these relationships if we looked at phylogenetic trees to see that. But there's a lot of things that kind of happen from this point to the point of us getting to our 5 kingdom system and our 3 domain system. And so that's kind of where we want you to spend a little bit more time reviewing, okay? In terms of, it's a very arduous task for us to learn every organism at each level of classification and so forth. So I'm not going to require you to like memorize the organisms at each level, the kingdom, phylum, class, order, family, genus, and species for several organisms. It's just kind of too much, and it's not a great use of our time. But I do expect you to understand how to use the classification scheme. So if I asked you different questions about, you know, what is happening at each level, or, you know, what something means at each level in terms of the similarities or relationships, or identifying, you know, would there be more members in one particular level of classification or versus another. I expect you to be able to use the information that we have here, but I don't expect you to know names of organisms at each level of classification. So the first example I use here was the human, because we're most familiar with the human. If we start thinking about the inclusion criteria, we know how to look for things like if we went outside and started digging, like if we took a trip to like Egypt and we started digging and we found like fossils of an organism and we weren't sure exactly what it was, we start looking for identifying features. You know, we have the skull, we have, you know, how many limbs are there, you know, are there, you know, a pair of eyes, you know, there are different inclusion criteria. So anyway, for the human, you've probably seen this alignment before. Obviously, we are in the kingdom Animalia, phylum Chordata, class Mammalia, order primates, family Hominidae, genus and species. You've certainly seen this scientific name before. Genus is Homo, species is Sapien, and so we collectively refer to humans. The scientific name is Homo sapien, right? So if I showed you some type of a schematic like this, I would show you this, and then I would ask you some questions, and I expect that you would be able to use the information in the schematic. We look over here at this orchid. Again, you know, I don't feel that it's a great use of time to try and go out and just remember classifications of all these different organisms. But again, if I gave you a schematic and asked you to identify the scientific name of this organism, what would you tell me?
Orcanis orca.
Yes. Yes. Orcanis orca. That would be the scientific name for this organism, right? And if I asked you to identify the kingdom or whatever questions I would ask you, I would give you the schematic and expect you to be able to use the information on it. All right, so let's keep going there. The last thing I wanted to talk about today is the process of science, right? Oftentimes we hear the word science and we hear the word biology and we try to say, or we assume that it's kind of the same thing, but it's not. Science and biology are 2 totally different things, right? And this is we're still in chapter 1 in your OER textbook. You do have a homework assignment tonight where you will read and annotate this section now that we're covering it, and it will appear backwards. This is actually going to be chapter 1.1. All right, we did 1.2 first because we identified these characteristics of life as we introduced biology as the study of life. But today we're going to pause and look at this concept of science, okay? a totally different subject or undertaking, right? So we're looking at the process of science. In your OER, it starts on page 27, okay? The process of science, this word science has an Latin origin, means to know, right? So I want you to think about science as a way of thinking, okay? We use science as a way of investigating the natural world around us, right? We use science as a means for systematic exploration of what's going on around us, right? So this in turn is a very broad subject, right? Biology is the study of life, right? But science is a tool, okay? Is a process for investigating. All right. And so you've heard about these things before. Science, again, is a tool. And so what is this tool used for? There's a very specific goal of science. Okay. What do you think that goal is? I just kind of alluded to it. What do you think is the goal of science as we define science? Go ahead.
As much things as possible.
I'm sorry, say that again.
To explain as much stuff as possible that we don't know.
Absolutely. That's exactly the goal. The goal of science, if I had to define it, I would say the goal is to give us precise information about the world around us. Okay. And so in order to get this precise knowledge about things, we have to have a systematic way of investigating, a systematic way of exploring and approaching our attempt to acquire knowledge, right? We understand that there are scientists all over the world that have this same goal. We're exploring something, we're trying to find out more information, we're asking these questions, we're making observations, we're collecting data. But in order for us to use this information in a reliable sense, we've got to have a standard for how we do this, right? And I'm sure you guys know where this conversation is going. And so what is the standard that we use for investigating the world around us as scientists? What's the standard? What's the standard procedure or process called?
Scientific method.
The scientific method. Thank you. The scientific method. Okay. The scientific method is in fact a series of ordered steps that we use to explore, to examine, to investigate what's going on around us. All right. So this is something that you've probably been hearing about at least since about the 5th grade or so, right? We talk about the scientific method, we ask questions, we formulate a hypothesis, we conduct our experiments and gather data, and we try to, walk away with some meaningful conclusion, all right? So we have to do just that now. Let's take a look at what is the scientific method and how it's super important in shaping our attempt at investigating what's going on around us. So we use the scientific method, right? The scientific method is a systematic way for us to explore what's going on around us. It starts with making some type of an observation. Our questions usually arise from an observation that we've made. We go outside and we see something that we might find interesting. We're immediately going to ask a question, all right? That now we want to answer this question because we saw some phenomenon happening and we want to know more about it. So we make observations, we ask questions, we develop a hypothesis, we then make predictions, right? Because we have these questions, and so with most questions, we can make predictions on what we think the answer to our questions are. But then we have to go out and actually conduct an experiment, do something that allows us to collect data. We collect this data, and then we have to actually sit down and analyze this data, right? We have to interpret our findings, okay? Data analyzation is a very critical part of exploring and finding answers to things, right? So we make predictions, we conduct experiments and gather data. We then interpret our results and we draw conclusions, Okay, and this is where we have sort of the most fun as scientists, because as we draw conclusions, we're now trying to expand these findings to develop theories and laws. Okay, so all the laws and theories that we hold to be true in science are the result of repeated experimentation, collecting of data, and analyzing and getting similar results over and over and over again, to the point where we can draw definitive conclusions from things. Okay, and so if you've never had to think about the scientific method in this regard before, we're now here understanding the scientific method as a systematic approach for science, right? Having now established the goal of science as obtaining knowledge, understanding the natural world around us. And it's very important that we understand that operative word here. We're talking about the natural world around us. We're not talking about things that aren't real. We're talking about real phenomenon that's going on, and we want to better understand them. Okay, what questions do we have? Any questions? All right, let's keep going. So one of the critical steps in the scientific method is formulating a hypothesis. Okay, this hypothesis is going to be critical to your investigation, and so there are some characteristics of a good hypothesis. Okay, when we conduct an experiment, we are essentially testing our hypothesis, right? And so before we get to that point, we want to make sure that we've developed a good hypothesis. So write this down. There are some characteristics that our hypothesis must exhibit in order to be considered a great hypothesis. Okay, one, it has to be reasonably consistent with well-established facts, right? So we're living in a world where, you know how people say there's nothing new under the sun. There's a lot of things that have already been well established, right? So as a scientist, you're coming out and you're asking questions, you formulate these hypotheses or you make these predictions. You can't just pull things out of thin air, right? You have to have a hypothesis that's reasonably consistent with things that we already know, right? Now, again, the goal here in your investigation, it is to find out new things, right? We don't want to conduct experimentation and investigate things that we already know the answer to. So yes, we want a novel question, but this hypothesis cannot be just a load of crock, right? It's got to be consistent with things that we already know. One of the main features of your hypothesis is that it has to be testable, okay? It has to be testable. We have to be able to design an experiment to which we can actually test what you are proposing, okay, as your hypothesis, okay? This statement has to be testable. And we should, whatever results that you get, it needs to be able to be repeated. Whatever results that we get from testing your hypothesis, it's got to be able to be repeated. It's got to be able to be falsifiable. We have to be able to show that we can disprove it. Okay. One of the things that we have to be careful about as scientists is avoiding bias. So again, we're scientists. We all have our areas of interest, and we have our, preconceived notions about things. We have an idea for what we want answers to questions to look like, but we have to avoid biases, okay? We have to design experiments that allows us to avoid biases. And let's look at some ways in which we can do that, okay? Scientists must do their best to avoid biases or preconceived notions when we're investigating. So I'm going to stop here though and give you a chance to chime in. Why do you think it's important that we design experiments that avoid bias?
And I mess up the experiment, like it won't give you the right data.
Right, because we don't want to, because what would be the risk of sort of setting this thing up to go the way that you want it to go? Like, what can be the bigger implications of that kind of thing?
You won't get the true result.
Like, your results can definitely be questionable, right? And the problem with that is that in science, we work as a body, right? We provide information to a greater body of knowledge, right? So what I'm doing in my lab as a scientific investigator, it doesn't just stay with me, right? The goal is to share my findings with the world, with everyone else. And so if I've sat over in my lab and designed all these things to basically say and end up doing what I wanted to do and say what I wanted to say, that poses a risk when you share that information, right? Because the scientists over in Europe can be reading about my experiments and use that as a baseline for the things that they're working on. And if my experimentations and investigations were not done with the most integrity, avoiding bias, with the proper controls and so forth, the results are not going to be as useful or reliable as you guys were saying, right? So one of the ways we do that, like you've probably heard about double-blinded studies, you've probably heard about things like placebos and so forth. But for example, like a double-blinded clinical study would prevent, say, the administrators or the doctors from knowing certain aspects of the study, right? So that we can actually get some true data being collected, right? If I don't know, for example, like say we're testing, just like even like with the coronavirus right now, it's a lot of conversation about the use of the vaccines and so forth. And let's say there is a drug company that manufactured this drug that we think might fight the coronavirus infection. We don't want to say vaccine. We'll just say a drug that you can take some drugs and whatever. And so during these studies, if you, as doctors, you're collecting data, you're writing down clinical symptoms and asking the patient about, how'd you feel? Did you have a headache? Did you have a fever? What temperature? Blah, blah, blah. And you've got a group of people over here who are taking the medicine. You got a group of people over here who might not be taking the medicine. But overall, we want to set up a situation where the physicians and the administrators and the healthcare providers cannot manipulate or influence what the results really are, right? So we set up these double-blinded studies where, again, the doctors are not privy to certain aspects of it. Their job is to just collect this clinical data, right? So they may not know if patient A or B received the actual drug or if they actually received the placebo, right? Because that can influence bias. Like if you think that patient A received the drug, now that may influence what you're reporting as a result of that medication, right? Anyway, so we want to make sure that we avoid bias. Let's stop right here. I don't want to overwhelm you with this idea of avoiding bias, but I want you to work on this for me before we run out of time. Oops. I'm going to share my screen with you. Oh, I don't think we talked about the controlled experiment. Am I missing a slide? All right, so we'll do number 1 in class, and I'll let you guys explore number 2 for homework. Let me share my screen. We have about, how much time remaining? Just about 10 or 11 minutes left in class. In your notebook, I'm going to share my screen with you. In your notebook, I want you guys to respond to question number 1, just based on the things that we just talked about. And for homework, I want you to work on number 2. I'm not going to let you turn in number 1. Just take a moment to respond to number 1. And we'll have a follow-up exercise on next class. But I mentioned the idea of the hypothesis, right? So this is a critical step in the scientific method. what are some characteristics of a good hypothesis?
Reasonably consistent with well-established facts.
You're writing it down. You're writing it in your notebook. You're responding to number 1 in your notebook, and then number 2, you'll explore that for homework.
Will you have this in a homework section sometime during today?
No, you have a different homework assignment that's already posted in Blackboard, but this is just for you to do in your notebook. But your homework that's in Blackboard, yes, it's under the homework tab. I'll get to that in just a moment.
So if you want us to do this in our notebook, how did you want us to submit it? Do you want us, or is there going to be like
No, you're not
submission
turning
box?
this in. You're not turning this in. This is in your notebook.
Oh, okay, thank you.
All right. So if you guys, if you're done responding to that number one question, I'll quickly mention the homework. If you look in your homework tab in Blackboard, and you can start on that now because we do have about 10 minutes remaining. I'm not going to add anything else to the lecture today, but you're looking at the process of science in your OER textbook, and that's going to be page 27 through 35. And you are reading and annotating and responding to the bullets that corresponds to unit 1.1. All right. So you're in your OER textbook here on pages 27 through 35. And this is going to do by midnight tomorrow night. You can use the rest of class today to go ahead and start on that if you would like. If you have any questions for me, you can definitely message me or, you know, stay back to ask them. Yes.
When we annotate those pages, do we need to send like a picture of our annotations to you at submission?
Yes, your annotations and your bullet questions are submitted for homework.
Okay, so if we like